Welcome everyone. Let's discuss oocyte transport. Let's have just a quick recap of the things which we have already discussed. In the previous lectures, we discussed how the oocyte is, the basic anatomy of the oocyte and the coverings of the oocyte. So, oocyte is surrounded by cumulus oophorus and zona pellucida separates oocyte from granulosa cells. There is something known as gap junctions, which we have discussed in detail in our previous lectures. Gap junctions provide the metabolic communication between oocyte and cumulus cells. LH surge leads to resumption of meiosis with oocyte getting arrested in metaphase of second meiotic division. That's why the oocyte is known as M2 oocyte. Just before the ovulation, disruption of gap junctions induces maturation and migration of the cortical granules to the outer cortex of oocyte. Oocyte and its cumulus mass gets detached from the follicular wall, leading to final release of the oocyte. The ovulated egg adheres with their cumulus mass of follicular cells to surface of the ovary. The fimbriated end of the tube sweeps over the ovary in order to pick up the egg. Fimbria on ovulating site are distinguished by being erect, probably due to engorged blood vessels. Entry into the tube is facilitated by muscular movements, bringing fimbria into contact with the surface of the ovary. So it is the active movement of the tube toward the ovary which facilitates the egg transport into the fallopian tube. Transvaginal endoscopic observation of actual ovum and cumulus oophorus pickup in women reveal that process is relatively slow, more than 15 minutes it takes. Let's discuss the normal anatomy and histology of the fallopian tube. It is important because for transport of the egg, it is the fallopian tube which plays the major part. It is not the egg, it is the fallopian tube. Active movement, active involvement of fallopian tube is important for the active transport of the oocyte. Gross anatomy of the fallopian tube involves, it is a seromuscular organ. It's a tubulo-seromuscular organ around 9 to 11 centimeter in length, and it has 25 finger-like projections which is known as fimbria. There are 4 segments from lateral to medial. First one is infundibulum, second is ampulla, then third is isthmus, and fourth one is interstitium. Embryology and development of the fallopian tube. From where the fallopian tube develops, it develops from the paramesonephric ducts. The most cephaloid portion of the paramesonephric duct develops into fallopian tube. The cranial end open and connect with the coelombic cavity. The caudal end communicates with the uterine cavity. Let's discuss the histology of fallopian tube. The fallopian tube has 3 layers. The inner mucosal surface, the middle smooth muscular layer, and the outer serosal surface, the serosal layer. The mucosal surface of fallopian tube consists of one layer of simple columnar epithelium which appears pseudostratified. The ciliated cells are mainly found on the apex of mucosal folds, mostly in fimbria and less commonly in the isthmus. Secretory cells are non-ciliated cells and contain apical granules that produce tubular fluid. Ciliary motility. Cilia extend from apical surface of ciliated cells into lumen of the tube. They are approximately 10 micrometer long and 0.25 micrometer in diameter. There is 9+2 arrangement in ciliated epithelial cells, while 9+0 arrangement in secretory cells. We all know that whenever there is a motility in any cell, that has 9+2 arrangement. The fallopian tube epithelium undergoes cyclic changes. Higher percentage of ciliated cells are seen in the follicular phase, while secretory cell dominant pattern is seen in the luteal phase of the cycle. Fallopian tubes are involved in a lot of physiological works. The most important one is mechanical transport of gametes and the embryos. The capacitation and storage of the sperms happen in the fallopian tube. The capturing of oocyte after ovulation is a work of tube which it performs actively. Maintaining and controlling the migration of spermatozoa to fertilization site. We already discussed this aspect in the sperm transport lecture, where we discussed how thermotaxis and chemotaxis are involved in migration of the spermatozoa to the fertilization site. The fallopian tubes maintain optimal environment which is required for fertilization to happen. They nourish the early embryo and they amplify the signals from embryo to mother, contributing to early stages of embryo development. What leads to egg transport in the tube? What are the factors which are important for the egg transport? First is the unidirectional ciliary beat in ampulla and isthmus of fallopian tube. Many cilia beat synchronously in the direction of the uterus. Fimbrial ciliary beat is faster in the secretory phase. The second factor which is involved in transport of oocyte in the fallopian tube is muscular contractions of the tube. Pregnancies in women with Cartagena syndrome indicate the importance of uterine and fallopian tube muscular peristalsis. Human fallopian tube muscular contractions are stimulated by prostaglandins E2 and F2 alpha and decreased by progestins, HCG and oxytocin. Tubal fluid plays an active role in transport of oocyte. Tubal fluid is very rich in potassium and bicarbonate and has very high concentration of arginine, alanine and glutamate. Maximal production of tubal fluid seen in mid-cycle to facilitate ovum pickup and embryo transfer. So all these three things are very important for the transport of oocyte by the fallopian tube. But with introduction of assisted reproductive techniques, the fallopian tube is being represented as an abandoned anatomical structure that is bypassed by IVF. In order to successfully achieve a spontaneous pregnancy, a complex system of tubal transport must be operational to allow a timely interaction between gametes and subsequently provide a conduit for developing conceptors to reach the uterus. Three different components: ciliary motion, muscular contractility and tubal fluid all contribute to varying degrees of effective tubal transport. To conclude, post-ovulation, the cumulus oophorus complex reaches the fallopian tube within 15 to 20 minutes. Tubal transport depends on three features: ciliary beat frequency, myosalpingeal contractility and tubal fluid. The fallopian tube provides an important holding action to allow time for endometrium to become receptive and blastocysts to become capable of implantation. A time period of approximately 80 hours, 90% of which is spent in ampulla. So thank you everyone. This was very important topic because the sperm transport and oocyte transport are very less talked about, but they form the basis for natural and spontaneous pregnancies to happen. And even in IUI, as long as the oocyte transport will not happen, the pregnancy success cannot come. So the emphasis behind this lecture was to show the fallopian tube patency is not the only thing which is going to work. It is the potency of the fallopian tube to capture the oocyte which is as important. Thank you everyone.